friends, this is Jyoti Saxena and I welcome you to my channel Biotechnology. Do you know the insulin used by millions of diabetic patients these days is produced by the microorganisms? So let's find out in this video how it is produced. In humans, the insulin is produced by the beta cells which are located in the islets of Langerhans in pancreas. The gene which is responsible for producing the insulin is located on chromosome number 11 and it is known as INS gene. The active insulin is formed by the proinsulin, and proinsulin is having a different type of structure than the active insulin. It is having 3 chains of peptides A, B and C. C is the middle one, and that has to be eliminated for the formation of active insulin. So active insulin has only 2 A and B peptide chains. And when we eat something, the blood glucose rises, and this is sensed by the beta cell. So that's how it starts producing the insulin, which goes into the blood, and it helps the cells to absorb the glucose. So that's how the glucose level is reduced and it is balanced. This is the structure of active insulin. Here you can see these are the two. This is the A chain and this is B chain, and here you can see the disulfide bonds, and these bonds are connecting these 2 chains. Now let's see how the industrial production of insulin started historically. So it started in 1923 by a biotech company, Eli Lilly, which is a very big company. And Banting and MacLeod got Nobel Prize also for this. Historically, insulin was extracted from the pancreas of cows and pigs. So that means many cows and pigs were slaughtered to get the pancreas. And at that time, the insulin which was extracted, that also was not very pure. Only after the isoelectric precipitation technique came, then the purification was a little better. And of course, the supply was very limited at that time. And according to an estimate, for 1 pound of insulin, around 8,000 pounds of animal pancreas was needed. So you can understand how many animals were, sacrificed for that. And that too was not sufficient for all the patients at that time. So the revolution in the insulin production came when the recombinant insulin was invented. So it was produced in 1976 by the Genentech company, but it was commercialized by Eli Lilly in 1982. And the first human insulin was named as Humulin by Eli Lilly. So let's see what are the production steps of recombinant insulin. First, we must identify the gene and isolate that particular gene. So mostly when we are isolating the gene, the pancreatic cells are used, although the genes are present in all the cells in human beings. Alternatively, the synthetic gene can also be used. That means chemical synthesis of the DNA can be done, which is very common these days. So that can also be done. Now the next step is insertion of that gene into a vector, because directly this gene cannot go into the microorganism. So first we need a vector, and here the plasmid vector is taken. And here you can see the picture. This is very crude picture. Here you can see the double-stranded circular plasmid vector, which is very commonly present in bacteria. And here this green color is showing the insulin gene. This is a very crude, very raw type of diagram. So just the location of the insulin gene is shown here. So this you can call as the recombinant plasmid. Then the third step is the transformation of the microorganism. Mostly the E. coli is taken, but alternatively yeast can also be used. E. coli is generally used because it is very rapidly growing. Every 20 minutes it can double, and also it is very easy to handle as compared to the other microorganisms in the fermenter. So here you can see the microorganism. This is the recombinant plasmid. This has to be transformed. This bacteria has to be transformed. Here you can see the plasmid has been inserted here, and this here you can see the bacterial chromosome, and here you can see the recombinant plasmid. So this recombinant plasmid is self-replicating body. It can replicate itself, and also it will replicate along with the binary fission of the E. coli cell. Once the transformation has taken place, the bacterial cells have to be cultured at large scale, at massive scale. So here this process is done in the fermenter or the bioreactor. Bioreactor is a vessel where all the parameters are controlled. For example, the aeration, temperature, pH, media. So everything is controlled. And here the E. coli, which has already been transformed with the recombinant vector, that is cultured in large scale and it is expressing itself, and the insulin is formed. So this is all taking place inside the bioreactor. Let's see the next step. That is the harvesting. Now, because the insulin is formed inside the E. coli, so harvesting has to be done. That means the lysis of the bacteria is done, and when the lysis is done, the insulin comes out, but at the same time there is lots of debris, cell debris is there, so it has to be separated from the cell debris. After the harvesting, conversion and purification comes. It depends on whether we have got the proinsulin or the A and B chains, peptide chains have been formed in different E. coli strains. That is also done these days very commonly. So if it is proinsulin, then enzymatically the C peptide chain is eliminated, and that's how the active insulin is formed. And then after that, the purification is done, and for purification different type of chromatography and many other techniques are used. And then after that, the formulation is into the medicine is done, because it is medicine. Insulin is as a medicine, it is used by the human beings, so the formulation is very important here. And it is available in rapid acting form or the long acting, short acting. So depending upon which category type of medicine is formed, it is formulated. Now let's see what are the major insulin producers. There are these three giants: Novo Nordisk, Sanofi and Eli Lilly. These are the companies which are responsible for making most of the insulin which is available worldwide. And other significant producers which are either making the insulin or the biosimilars are China-based and Germany and US-based companies, these three companies. And there is one Indian biotech company also, Biocon, which is making insulin as well as biosimilars both. Now the question is, why recombinant insulin is better? The first reason is this is identical to human insulin. It is, even the cow insulin and the pig insulin were not identical. They were close to human beings, but not identical. So this one is identical, and very few allergic reactions have been recorded as yet, because millions of people are consuming this, and it is safer. And of course it is very pure these days. To make the insulin very pure or purify it, many different techniques are available, so it is quite pure and it is accessible. It is very readily available in the market, so everyone can go and buy it. And it is also affordable, because I told you that animal insulin was very expensive, because many big animals were slaughtered for that production. It was impure also, so it was very expensive, but this one is affordable. So at last we can say that no more animal slaughtering is done these days. This insulin which is available in the market so readily, that is genetically engineered, and first we genetically engineer the microbes, and that's how we get the insulin, which you can call as the recombinant insulin. And one of the most successful example of recombinant DNA technology transforming medicine is the production of insulin. So this was the first product which was formed with the help of the microorganisms. using the recombinant DNA technology. So you can see how the recombinant DNA technology is bringing the revolution in our lives. So that's all for this video. Thank you very much.